Hello, my name is Sadie Holt, and I'm an undergraduate research assistant in the Bowerman Sports Science Center at the University of Oregon. My project investigated the effects of mild hypohydration on acute kidney injury biomarkers in healthy young adults. But why is this important, you might ask? Well, recent data highlights that when an environment goes through a heat wave, as is happening more and more due to climate change, there's an increase in hospitalizations due to fluid or electrolyte imbalances and acute kidney injury. Acute kidney injury can be measured using biomarkers, which are proteins that can be found in the urine and indicate some level of stress on the kidney. Current data suggests that between 33 and 74% of people in the United States are chronically mildly dehydrated, which increases the severity of the dehydration they might experience as temperatures in their environment rise. There is a knowledge gap in whether acute kidney injury biomarkers are present during a state of hypohydration, which is a state of low body water, when this occurs independent to heating on the body. So our research aimed to bridge this gap to further understand the pathology behind the increase in hospitalizations during a heat event and to provide accurate countermeasures for these individuals. So let's get into the study. Participants were comprised of 22 healthy young adults between the ages of 18 and 24, and they were randomly crossed to perform either the euhydrated trial, which is a normal hydration status, or the hypohydrated trial first. In order to induce mild hypohydration, participants refrained from drinking any fluids or consuming high water content containing foods for 24 hours. Participants then performed the opposite trial with a minimum of 72 hours washout between the two. After participants performed the protocol for 24 hours, urine samples were collected to measure the presence of the AKI biomarkers, or acute kidney injury biomarkers. The analysis was completing using ELISA assay kit, and 4 important biomarkers were measured due to their current clinical application and their indication of stress on specific anatomical areas of the kidneys. So these biomarkers were the product of insulin-like growth factor binding protein 7 and tissue inhibitor of metalloproteinase 2, or IGFBP7 times TIMP-2, which is a biomarker indicating renal cell cycle arrest that has the only FDA-approved marker for AKI risk screening. The second biomarker measured is kidney injury molecule 1, which is a marker of proximal tubular injury. And the fourth biomarker measured is neutrophil gelatinase associated lipocalin, or NGAL, which is a marker of general renal tubular injury. So moving on to our results section, I'd like it to take a minute to orient you to the figures. So on our x-axis, you can see here, our participants are shown as a group of 22 and then deaggregated by sex. Black bars indicate the hypohydrated condition, and white bars are the euhydrated condition. Circles indicate individual male data points, and the squares represent females. The results of the ANOVA table are presented on top of the figures, and exact p-values are for post-hoc multiple comparisons are shown directly above a given comparison. So starting with the graph A, this graph indicates the hydration status of our participants, which is shown with a larger decrease in body mass during the hypohydration state, consistent with our expectations for the dehydration protocol that we had the participants follow. Graph B indicates a marked increase in the AKI biomarker IGFBP7 times TIMP-2, which, if you remember from previous discussion, this is our only FDA-approved marker for acute kidney injury risk. So the hypohydrated group had higher levels of this biomarker without any heating due to their decreased fluid intake. Graph C shows a similarly significant increase in the biomarker KIM1, indicating acute injury to the proximal tubule during hypohydration, independent of heating as well. And for graph D, there was no difference in the NGAL biomarker presence between the two trials, but the unexpected finding is between sex differences, with a higher level of the NGAL biomarker in females when compared to males at both the euhydrated and the hypohydrated state. And this will be a fantastic area for future research to be done in. These data overall support our hypothesis that prolonged mild hypohydration increases biomarkers associated with renal epithelial cell injury that suggest increased risk of acute kidney injury. This conclusion is vitally important in understanding one of the causes behind the increased hospitalizations during heat wave and how we should move forward in advising people to protect their health prior to a rise in temperature. Thank you. I'd like to thank my advisor, Christopher Chapman, and all the other students who assisted with this research from the Bowerman Sports Science Center within the Department of Human Physiology. Thank you.